Okay, so hello, good afternoon, everyone. Can all of you hear us? Hear me? Sorry. Okay, thank you, thank you. So welcome. Good afternoon. Welcome to the first open lecture on the introduction of synthetic biology. Today, we are going to share our passion, our joy, and interest in the field of synthetic biology. And we hope you guys can share this passion with us. And maybe we can get you on board and we can do some synthetic biology together in the future. So first thing first, who are we and what do we do? So yeah, we are HKU iGEM Team 2020. I'm Joe. I'm here with Sophie, here, Munisa and Eric. We are all team members of the HKU iGEM team. So iGEM is short for International Genetically Engineered Machine Competition. So what does it entails? We actually use organisms as a sort of a chassis or a vehicle to engineer through the genome and carry out different functions and different activities we want. Yeah. So here you can see the pictures. Yeah, the photos of the iGEM competition 2019. So these are our team last year. So iGEM is an annual competition. It's a synthetic biology competition where there are over 300 teams over around the world. They develop solutions and developed different devices and different solutions to solve different problems in the world. So annually, we will present our results to judges around the world in Boston. So here are just a few of us presenting in the giant jamboree in Boston last year. So yeah, and so whether or not you have the science discipline background or whether or not you have biology background, synthetic biology is for everyone. So whether you are from science, medicine, or law or business, just like this year HKU team, we are fortunate enough to have such a diverse background of students from ranging from engineering to medicine to science to even business and politics and public administration. So enough of the introduction of ourselves. We will jump right into today's main topic, synthetic biology. So as Ian Malcolm from the Jurassic Park said, life finds a way. As harsh as the environment and as adverse as the condition can be, life finds a way to get through all of this and to generate different solutions, different innovative methods to bloom and develop into a vast diversity of life as we see today. So I will pass to my teammate Eric to tell you all about basic biology and a brief introduction of synthetic biology and the history. Thanks, Eric.
Hello, everyone. This is Eric, and I would like to start from the origin or the history of life on the Earth. So here I demonstrate with a evolutionary timeline from the beginning, where the Earth formed, and as we are at the point where life is very diversified and exists as nowadays. So starting from the Earth formation, we note that it wasn't really hospitable condition for life to be sustained. And as time passes, Earth cooled down. And from 4.2 billion years ago, we noticed that some kind of inorganic chemicals started to become organic chemicals, which was actually demonstrated in the Miller experiment, where he provided some energy input to create organic molecules, such as the basic unit of life, amino acid, from inorganic compounds. And also moving on to the next, which is a most well demonstrated hypothesis to explain the first stable molecular life structure is the RNA, and we call it RNA world hypothesis. Well, RNA is the best candidate for the first stable molecule of life because it carries both genetic information and it also acts as the catalyst. So, and from there, phospholipid bilayer formed to shape a cell, and which kind of distinguish it as a different entity, so that it can be very efficient in carrying out the gene material, and it also stores very efficiently. And then we call that LUCA, which stands for Last Universal Common Ancestor. So this is where current scientists see as how life originated, and it starts to diverge into different types of or forms of organisms to fit the environmental burden that they have. So moving on to the next slide. So I would like to talk about the fundamental structure, as known as DNA. Its shape, as you guys know, is a double helical structure, and by having this structure, it's very stable in storing the genetic material. It composes of the sugar backbones, and also it has the 4 different types of base pairs, which will later encode for the protein. And here on the right, I demonstrate the concept of central dogma, which is how the DNA will be transcribed and translated so that we can make different types of proteins for different functions. Moving on to the next slide. So here is the transcription, which is a process by which the genetic code contained within a messenger RNA molecule is decoded to produce a specific sequence of amino acid in a polypeptide chain. And oh, actually that was translation, my bad. So transcription is actually, it forms the mRNA, which will later be decoded into protein. And RNA polymerase is a protein that is used in transcribing. And moving on to the translation. So here, like I explained, it produces a specific sequence of amino acid in a polypeptide chain. And it occurs in 3 steps using tRNA. I'm starting with the initiation, where the first tRNA with the protein amino acid attached to it binds to it, and also it elongates, and at the end it terminates and having the whole structure of amino acid. Okay, and the table here explains the importance of sequence and the combination of how the mRNA sequences work together to produce different type of amino acids. Okay, so to me, it's very interesting to see how life becomes complex as time passes. So, which is conflicting with the concept of second law of thermodynamics, where the entropy only increases as the time passes, which we may see it as a commonsensical concept when we see the universe, but life actually works in the opposite way. So, in the evolution of life form, species adapt to the changing environment, and they will have a different kind of kingdoms, as shown in here, to suit their best capability for sustaining their own life. So, moving on to the next. So life in general is complex, but it could be fundamentally explained with logic, and thereby we can call it as a process of engineering. And it's very systematic in a way where scientists could tackle in and do more things with it. So with this idea, we can move on to the idea of synthetic biology, which I ask you guys a question first. What do you think synthetic biology means? Okay, I guess I'll just okay, making biological products. Yeah, quite similar. Okay, so I'll give you the definition now. So synthetic biology, by its definition, states as a multidisciplinary area of research that seeks to create new biological parts, devices, and systems, or to redesign systems that are already found in nature. So synthetic bio is a field of redesigning organisms so that it can meet the purpose of what engineers wanted to have. And many researchers and companies around the world are harnessing the power of nature to solve problems in medicine, manufacturing, and agriculture. So some of the examples may include microorganisms harnessed for bioremediation, which is to clean pollutants from the water, soil, or the air, or some other field, which somewhere in an agricultural field, rice will be modified to produce beta carotene for people with vitamin A deficiency. Also in other field, it can be used for engineering yeast to produce rose oil and substitute for the real roses that perfumers may use for the luxury scents. So as you can see, it's a very diverse field we can tackle through biological systems. Okay, let's move on to the next slide. So here I break down the synthetic biology in a literal meaning, word meaning. And here synthetic refers to man-made or something that is artificial, where biology, on the other hand, refers to the life, which is something that happens naturally. So this is, in sense, seems very paradoxical, but trying to utilize the natural existing bioparts to build into something intentionally produced could provide a huge potential in this contemporary world to solve many kinds of naturally existing problems. Here I also want to point out one thing where people may mistaken the synthetic biology with the concept of genetic and genome editing. In synthetic biology, scientists typically stitch together the long stretches of DNA and insert them into an organism's genome. And these synthesized pieces of DNA could be genes that are found in organisms, or they could be entirely novel. However, in genome editing, scientists typically use tools to make small changes in organisms' own DNA. So here we have a similar in terms that we are manipulating DNAs within the organism, but how we tackle this is quite different. Okay, so next, moving on. Oh, okay. So here I explain the history of synthetic bio. Here, first, Gregor Mendel discovered the gene, and the concept of law of segregation was the first founding that happened. And then Watson and Crick found the structure of DNA to be double helical, and next, Sanger developed the Sanger sequencing technique to actually sequence the DNA. And then recently, Jennifer Doudna developed the CRISPR system, which we can actually edit the gene from inside the organisms. So here's the brief history, and moving on to the next slide. So Within the completion of the Human Genome Project in 2003, along with other organisms, we have developed a new sequence attack called next generation sequencing. And it led us to have the gene sequencing costs much lower, as you can see from the data on the right. And also, on the other hand, the genomic data amount skyrocketed. And this is the end of my part. And now on, Sophia will lead on with the details.
Hi guys, can you hear me? Right. Hi, I'm Sophia, and I'm going to tell you some of the basic concepts in symbio. So unlike biology, synthetic biology is actually more like an engineering study, but it happens in a biological system. So synthetic biologists standardize the process to manipulate genes and create new biological system, or redesigning the current one. So the list on the left are the symbols or codes for synthetic biology. So basically, there are different kinds of elements, and scientists often need to characterize the parts by knowing the properties, such as the binding efficiency or the half-life of the protein. So as I said, synthetic biology is a combination of science and engineering. So it also requires the techniques and the concepts on both sides. In science, as some of you may know, that we need to develop hypotheses from observation, followed by prediction and experiment and finalizing the theory. While in symbio, we also mix with the principle in engineering. So we design a system, building it up, testing or validation, and learn, which is just like doing some modeling for the system. So these are some of the illustration on the principles of engineering and science. And so in doing synthetic biology, we first need to identify the problem and hypothesize where the problem is, and design a device or method to tackle the problem. After we have the design, we build it up, just like building some genetic circuits. And then we test the system with some experiments and also identify the weakness of the system. And then we try to apply it to the problem that we identified and try to tackle the problem. And eventually we come up with a theory. Well, as I've just said, that synthetic biology is a multidisciplinary subject. So in this slide, we're showing an example of biosensor, and we want you to think of any subject such as biology that is involved in this case. So in this system, lexapron encodes for the enzyme that produces light when it's placed after a promoter that is responsible to that can respond to environmental stimulus, such as petroleum pollutant. So we create a biosensor by coating a bacterial layer on a photosensitive computer chip. Can you guys think of any subjects that are involved in this case? Comment. You can unmute yourself, or you can leave some comments in the text in the chat section. Do you guys have any idea? Oh, I'm talking about like do you know any discipline that can, any subjects that involve in this case, for example, like physics, computer science, etc.? Oh yeah, of course, physics. And can you think of more? Yeah, true, Betty, true. It's computer science and also, of course, yeah, chemistry as well. But to be more specific, it should be biochemistry because it's a bacterial reaction, bacterial chemical reaction, right? So let's move on then, if there's no more response. Okay. Right. So one of the brilliant things you can do with synthetic biology is that you can do fancy things with simple techniques. All you need to have is some basic knowledge in different area. And what's more important is actually your creativity. So I'm going to show you now what a synthetic biology do in their everyday life. And I'll take iGEM team as an example to explain some of the essential wet lab techniques. So as you can see, these are the photos of our iGEM team doing the wet lab job. Yeah, it's very, they were having fun, I guess. So for iGEM, we deal with a large amount of bio bricks, and they're like Lego. We just need to design the architecture of the genetic circuits, select the appropriate bio bricks, process it, and stick them together. So if you look at here, this is a standard structure of a bio brick. It usually comes in a plasmid, and this is the bio brick part. It contains a gene or genetic element such as the promoter or terminator. The backbone will usually contain sites of restriction enzymes for the cleavage so that the bio brick part can be cut out and stick to the other bio bricks. So in this case, in the standard iGEM bio bricks, it usually comes with 4 restriction enzyme sites such as EcoR1, SBA1, SP1, and PST1. So how we put the bio bricks together is by a technique called molecular cloning. We first use restriction enzyme, just like a scissor, to cut out the bio bricks or the gene of interest and the factor as well. In this case, it's a bacterial plasmid. So the cleavage will generate sticky ends, which helps further ligation by the DNA ligase. And the final product is recombinant DNA plasmid. So in actual life, synthetic biologists just repeat these steps multiple times to put different bio bricks into the same plasmid. So after we finished cloning our product, we will transform it to bacteria, usually by the method of heat shock. Basically, it just make a small recoverable hole into bacterial membrane so that the DNA could slip in through the hole. And since the plasmid contains antibiotic resistance, the bacteria with DNA transformed can grow on an agar plate here, the yellow part, the yellow plate, and containing antibiotics. So the bacteria will grow and eventually form a colony, just like what we see here, the white dots. And the organism with recombinant DNA, the organism with recombinant DNA usually express the phenotype immediately. For example, the bacteria transformed with plasmid containing human insulin genes produce insulin, which is a major manufacturing source of insulin these days. And you can see that there are some transgenic mice generated by micro-injecting fluorescent genes into the genome. So they show some green fluorescence when they're in the dark. Yeah, as I've just said, that synthetic biology is not just a biology, it also composed of different kind of disciplines. Right, that these includes engineering, mathematics, biology, and chemistry. And we require these knowledge to characterize and rationalize the chaotic nature of life. So to make the symbio impact the world in a more profound way, we also need other subjects such as business, ethics, or philosophy and law are also essential to the progress of symbio. And to tell you more about that, I will pass on to Munisa. She will show you some modeling techniques.
So hello everyone, my name is Munisa and I will show you some progress and recent advancements of synthetic biology in terms of using different computational tools to create data and analyze the samples for aiding the progress of synthetic biology projects. So in a lab setting, we usually perform experiments with testing different circuits and performing some time data analysis experiments. And usually we need a lot of help to automate some processes and generate some predictions in order to better design our experiment and to analyze its output. And that's why synthetic biology is such a great area, because it's not just handling the live things in the laboratory, but also designing and improving synthetic tools from different experiments in order to predict it better. So engineering of life indicates accurate prediction of all sorts of behavior in life, and extensive modeling is hence needed to compare expected and real life behavior. So as you see in the picture to the right, we can start from showing the biological systems, and these biological systems can be predicted by different mathematical models. So these mathematical models can predict the behavior of different molecules like DNA or some proteins and their interaction. So for example, the DNA polymerase binding to the DNA for expressing the proteins we need. So these kind of mathematical models can be designed, edited, and improved by using synthetic biology tools. Computational design doesn't even end in this. Instead, not only we need to explain the behavior of our models that we develop, we also need to predict the future occurrences and generate potential variables we will need in order to design our experiment better. So synthetic biology is also very connected to metabolic engineering. So these are the computational analysis of metabolic networks and the engineering of pathways. So as we know, a living cell consists of millions and trillions of different pathways of chemicals. Chemicals dynamically change their behavior and go from one state to another. And synthetic biology helps to make all these data quantitative in order to better design and predict the behavior of the systems in biological organisms. So as you see at the bottom of the figure, there are different parts already constructed and characterized using synthetic biology tools. And one of the advantages of Synbio is that everything is characterized and automated and available in open source. So let me show you some of the tools that are helping us synthetic biologists nowadays in order to advance their field and do better research. So one of the really raw tools that synthetic biologists use that are easy to learn and the learning curve is low is programming languages like R and Python. And these languages are kind of not very, their syntaxes is easy, resembling almost an alphabetical language. And they are flexible enough to perform different tasks that can help scientists working in the wet lab to better design the experiment. One of the examples is to use different languages. In my example, it's Python because Python is easy to use in order to find the replication origin in one of the bacteria called E. coli. So replication origin is just a part of DNA that basically is recognized by some molecules in the cell, in the bacterial cell, in order to know where to start replicating. I'm sorry for the background noise. Yeah. So as you see here, we have a list of nucleotides over here, and there are some certain regions that are highlighted as brown and green, and these regions are are a sequence of letters that represent nucleotides of DNA, and these sequences is, if you can see, kind of resemble each other. These are repeats that are used by scientists working with computers in order to recognize where this origin of replication is located. So because the genome of organisms is very long, it can consist of 200,000 nucleotide base pairs. It's very hard for, without the aid of computational tools to find this replication origin, and knowing this location is very important in designing experiments. So what can be done in this case? So usually in the early rise of bioinformatics, scientists just use simple languages like Python in order to create some codes that I can show here. So these are the codes that recognize and read the DNA. So these different letters that are like live organisms alphabet, and they recognize all these letters and try to locate the window where that potentially can be the origin of replication for this particular organism. So as you can see, these synthetic biology tools can help scientists a lot, since performing the wet lab experiment can be tedious and long, whereas with these short, relatively short code lines, it's possible to pinpoint the potential locations. So Python and other raw programming languages is just one of the examples of let me show it here. So yeah, it's just one of the examples of what synthetic biologists use nowadays. Since the field has risen in bioinformatics and other interdisciplinary sciences related to life sciences have evolved, there has been developed some programs that are that have a very nice interface and are used routinely by scientists in the wet lab. So one of them is of course SnapGene, which is just a software that is available, that's available almost to everyone, and people can use it to design their DNA fragments and perform and kind of like simulate different experiments like restriction digestion or cloning procedures. Next slide is here. So some of the since we have just right now talked about the computational design for forward engineering of genetic circuits, biological systems, or cells. There is also another part of hardware and software tools that that can aid the progress of synthetic biology. This is basically analyzing metabolic engineering pathways and trying to predict their behavior and developing complex circuits that can be expressed quantitatively. So one of these softwares is iGEM, and this is just one of the examples of softwares that can design a circuit that you want. Basically, here you can design the circuit that you want. You can generate a model, and basically you can have an analysis of the circuit behavior between each other. So if you can see here, this is one of the circuits that I designed. These are all the parts that can be found on the internet and inserted in here. So everything is connected. The parts, their genetic sequence, the part name, and the part location on certain servers that are open source, like synbio-focused. They are all connected in order to kind of integrate all the knowledge and experience of other synthetic biologists in order to kind of like fast forward the progress. So basically, after the designing of all the circuits, we can basically establish relationship between different kind of molecules and outputs. So for example, here, during the production of certain molecules, there is inhibition or activation, and basically later on you can develop a model and generate a graph in order to visualize and simulate all the different parameters that you want to use. So so where is the presentation? I think it's in the computer over here. Ah, never mind. I think I can do it myself. Okay. So basically, that's it. This is one of the reasons why synthetic biology is an emerging tool, because various people outside of the field have been very skeptical about everything that is related to life sciences, because there was this misconception that life sciences are hard to predict and hard to mold to kind of serve your aims and goals. But nowadays, with the development of so many softwares and tools, it's kind of really easy to develop complex circuits, analyze them, and basically reach your goals easily, and do this incremental progress of the field. And let me next pass on to my friend so that he could explain so many different applications that synthetic biology can offer and has done tremendous achievements in.
Okay, okay. Yeah. Okay, thank you, Melissa. So moving on to the next slide. Sorry, wait a minute. I can share. Sorry. Yeah. So moving on to the next slide.
Okay, I'm so sorry, I'm muted. Okay, so as Monisa said, there are many tools and software built by scientists to increase the accuracy to predict the biological system. There are many research been going on to adapt a genetic system into be like mimic the electrical system. So for example, in computer science, there are logic gate, like AND gates or OR gate. This could actually be built by using different genetic parts. Here, we can use different multiple inducible promoters and use a reporter genes like called GFP, which is short for green fluorescent protein. By inputting different inputs, different chemicals, we can actually control the biological system to producing different outputs. The other application is that, yeah, there are a oscillator. So in engineering, an oscillator is a system that kind of produce a signal in a periodical fashion. There's a low signal, there are high signal, no signal, high signal, etc., etc. So we could actually build this using genetic system. With the first design of this genetic oscillator is called repressolator, which the scientists use 3 different repressible promoter and to interact with each other and to repress each other in order to periodically express the GFP protein. As you can see here, here is a animation of the oscillator system in action in bacteria. Yeah. Okay, so moving on, there are many different applications in synthetic biology. We will just cover a tip of the iceberg of the application here. So first of all, obviously, there are many applications in medicine. So for example, there's a US-based company called Synologic. They engineered a microorganisms. They engineer a microbe that could detect a toxic buildup of a chemical called phenylalanine. So when they sense the concentration of phenylalanine is too high, they will switch on the genetic circuit that is placed inside them and to break down the phenylalanine. This is actually a rise of the era of living medicine. We can use microorganisms or other living system as a therapeutic solutions to tackle different health problems. Also, in the production of energy, because the fossil fuel is like deteriorating, we need more reproducible energy. So there is a science team to use an interesting bacteria called geobacter to produce energy out of thin air. So this geobacter actually contains a very fine, very small protein nanowires in their bodies. So when they these nanowires can actually transport electrons within each other. So scientists develop a thin film they use with these nanowires to produce the electricity with the difference in humidity. So you can place this film everywhere to generate enough electricity, like to power your phone or etc. And also, there are some interesting research going on in the construction area with synthetic biology, as scientists can now build a so-called self-healing cement with the microbes built in. So the concept is that the bacteria have the metabolism to produce limestone using calcium. So we mix this bacteria into cement, and a so-called bioconcrete is created. When there is a cracks, like there is a damage in the cement and there is a cracks, the water and air comes in and make the bacteria active. After that, it will metabolize calcium and make the cement like grow again. So the cement will self-repairing. Also, in the field of fashion, yes, fashion and synthetic biology harnessed by different designers and different factories to produce a sustainable materials using microbes. So here you can see a founder of the biofabricate, Susan Lee, which talk about how to talk about the fourth industrial revolution, where we use living things to create different system. So on top of that, on top of this real life application, we human look beyond the future and look. We want to build a completely new biological system to maybe do some more research on the origin of life or sustain ourselves long time in the future. So there in synthetic biology, there's a concept called bottom-up engineering. So you build a new organism from scratch. Either the genetic code, the DNA, the cells, all of them is artificial and man-made. And actually there is a new DNA structure called XNA, which is a completely new design of the DNA, which can surprisingly function like the actual DNA. And they also expand the genetic code, like except for ATCG, there are also P and Z, so they can expand the genetic code and build more proteins or functions in a natural biological system. And also there are research on de novo protein design, which so scientists can build different system using like computer and creativity. Yeah. So as like imagine that we want to put man on Mars or go to other planets on other solar system. So there is a plan to Mars already in place, but what can we do? So what should we do to live and succeed in outer space, like far, far away, like few light years away? The first problem is that for 1 light year, it takes 20,000 years to arrive, even with a very, very fast spaceship. And for Mars, it took about 7 months. So the longevity of human life is just not enough. So there are research going on to like improve the longevity or enable the human to live in a cryoprotective case. And another thing is synthetic biology can also be used to terraform a planet, like a lifeless planet like Mars, because Mars, there is not much water or nutrients to support the life on Mars, because we need food. When we arrive in Mars, we need food to live, and so we can use the synthetic biology to grow food and the terraform Mars into a more habitable planet. So this is a like ideal future of our planet or other planet. Yeah, and so last but not least, because synthetic biology is talking about tempering the very nature of the life itself. So there are many questions around the ethics of synthetic biology. For example, people suggesting to democratize biology. So as the cost of research of gene editing or genome sequencing keep plummeting, there are increasing people not in the academia or industry. They do science, they perform experiment in their home. They are so-called biohackers. So this could potentially accelerate the biological research, but still, if they release some engineered organism accidentally, this will cause a devastating result, such as the gene drive. This is a technology where you put a CRISPR gene into a germ-like cells like egg and sperm. So over generation, this organism can, because the offspring contain this gene drive gene, cannot reproduce, and so over generation, the whole population will be wiped out. So based on this research, we can most of the scientific research is like a two-edged sword. So if you scale the regulation to either edge, either side, so you will have a under or over regulation. So for example, CRISPR can be used to eradicate genetic diseases, but it can also like commit a genocide on a group of people, like using gene drive to kill a certain race of people. So if we ban any CRISPR research on human based on the harm that can do to people, so genetic disease will persist and the research will be like stopped and no process ever. An example is a Chinese scientist called Hao Jianhui. So he created a CRISPR baby. It's also like a stir and hot debate over we can use human gene editing to like edit babies and change human behavior, nature, and stuff. Because of his illegal human experiment and his reckless experiments on humans, an international moratorium on gene editing is actually in place. So in US or in some country like Germany, the gene editing in animals, in human is actually prohibited. So in these countries, the research on human gene editing is not good. Yeah, so we think a code of ethics should be created and educated to all scientific researchers, be it academic or commercial or hobbyists. A code of ethics should be created to tell them how to behave as a scientist. So for your information, there is a very good documentary about a pros and cons and future of gene editing in synthetic biology, which is called Unnatural Selection. You can check it out on Netflix. So last but not least, this is just our first SynBio lecture, a pilot test. We want to package it, we want to promote it, package it as an unconventional science courses here in HKU. We would like to constantly provide or challenge our students with advanced, recent advancement in the in recent years, instead of repeating and regurgitating a research from half a century ago. So yes, this is our vision of creating an open SynBio lecture. So we can keep challenge ourselves, and you can we created some kind of resources for you to, some kind of resources for you to check out, so you can see more about synthetic biology. Wait a minute, I can always share to everyone here. Where is it? Sorry, wait a minute. Here. So here is, I uploaded a PDF and you can check it out to see more about synthetic biology. So, yes. And yeah, you can, synthetic biology is also not only about the knowledge and stuff you learn from school. You can do a wide range of search on Google and many different websites or YouTube or Netflix, just to name a few. You can do your own research and to know more about synthetic biology. So we actually have a general syllabus in plan. We will not stop in this first lecture. We want to make synthetic biology an official course in HKU. Maybe through an open lecture on platform like edX or like open course, like online course. So here our general syllabus in plan. If you are interested, you can contact us and leave a comment and send us any feedbacks and challenge us, question us to make a better lecture for more people alike. So thank you for attending this first lecture of synthetic biology, and I hope you enjoy this first lecture as we do. And here, and here you can contact us through this platform. So thank you. And if you have any question, you can ask us now or afterwards. Yeah, thank you.
There's a question. How can we engineer a genetic circuits in the lab?
So sorry, wait, where?
Wait,
Okay. Okay, I see. How can we engineer genetic circuit in the lab? Okay, so based on the knowledge we have, like different characterization of basic biological parts, like promoters, the ribosome binding site we call RBS, the protein coding gene, the terminator, and all of these parts we can you can search on a website called biobricks.org. Yeah, so you can search different parts. So like for say you want to build an oscillator, so you will need some, we have some inducible promoter, some protein coding gene, and some reporter. So reporter is for you to like report the states of your biological system. So after searching, after like designing the system and searching on Google and stuff, you can, if you want to know how to build a genetic circuit from scratch in a wet lab setting. So actually, so Sophie tell you about like how the cloning stuff works. So because like after you build an oscillator, like the full restriction digestion and put the Legos components together, you can build it. After restriction digestion, you can put it in bacteria and express it, and you can see the genetic circuit in lab. So does this answer your question? And okay, and Betty, right? Yeah, you just said the biological system is complicated and quite difficult to be fully controlled. So how can scientists minimize errors in synbio? So as Munisa said before, before we perform a real-life experiments, we perform different mathematical modeling with software and program, and so we can model how the biological system will behave. So after that, after you get all the components together in computer, you can perform experiments in a controlled manner and compare the experimental result and the result from your mathematical model. So if they match, so it means you are onto the right thing and to minimize error. Yeah.
And also I want to add on. Yeah, I guess, yeah. Can you hear me? We should mute yourself, I think.
Okay.
Other than that, I want to add on how to make sure the safety issue is applied. So let's say that the errors were unavoidable, and there are some ethical and safety issues when you're trying to engineer a living organism. So there are certain standard procedures that usually synthetic biology scientists in the lab carry on in case there will be some errors, and there will be some errors in like some mistakes in containment of the biological systems inside the lab. So to avoid this kind of issues, there are certain constructs that synthetic biologists add on the circuit apart from their circuit of wish. So there are some systems like kill switch where there are certain toxins that are added on some certain parts that that are added on the circuit, so that if there is some mistake inside the biological organism, there will be a way to kill the bacteria and avoid the containment problems.
Okay, so do you guys have any more questions? Yeah, thank you. Thank you. So if you want to ask more questions or reach out to us, you can find us through Instagram or Facebook or email us. Thank you very much for attending this lecture. Goodbye, guys.
See you.
Thank
Bye.
you.
I'm going to stop recording.